Morning, everybody. We'd like to get started. Um, Welcome back. This we have upcoming lectures, grand round lectures that I think that will be very exciting. On the 18th, which is next week, Holly Lisenby, who is the director of translational research, the Division of Translational Research at NIMH and the former chair of psychiatry at Duke University, will be talking about, her title is Precision Seizure Therapy, Sculpting the Temporal and Spatial Aspects of Therapeutic Seizures. And Dr. Lisenby has been a real pioneer in electroconvulsive therapy research and also developing a new approach called magnetic seizure therapy for treatment-resistant mood disorders. And she's a wonderful scientist and a great speaker, and it should be a good grand rounds. The following week, on the 25th, in a stimulating doubleheader, Nolan Williams, who's a professor of psychiatry and neurology at Stanford, will be talking about interventionalism in psychiatry. What can we learn from cardiology, neurology, and radiology? And he is someone who is involved in a number of areas in neurostimulation, but including deep brain stimulation and transcranial magnetic stimulation in particular. And so should be, both of these people are very accomplished and should be wonderful grand rounds. I'm thrilled today to introduce our speaker, Joy Hirsch, who is a member of our faculty and whose laboratory is on the 9th floor of 300 George Street in the Haskins lab space. And so if you become inspired from the presentation today, I highly recommend looking her up. But Joy is really a pioneer, both technically and in many areas of cognitive neuroscience and neuroimaging. We were extraordinarily fortunate that she chose to move up to New Haven from Columbia, where she had established and led an MR neuroimaging research center for a very long time and pioneered many areas of cognitive and clinical neuroimaging research in that capacity, and also served as a mentor for a very large number of very accomplished people who are now leaders in the field, people like Amit Etkin and others. And her contributions really, and her honors are really too many to list, but they range from really diverse areas like autism or studying brain activity in people who have locked-in syndrome or comas or other clinical psychiatric applications, but also basic sort of cognitive neuroscience. When we were fortunate to recruit her up to Yale, she established a very unique program of research and was able to partner with an industrial maker, someone technically innovating, to create, to employ a technology called near-infrared spectroscopy, or NIRS, which is a new, relatively new neuroimaging technique that she will describe in more detail, I'm sure. But she, at the outset, set her aims very high to try to understand how people interact with each other, an emerging area called social cognition, or social cognitive neuroscience, really. And so that she's recording the activity of the brains of two people as they're interacting with each other to really help us to understand the dyad as the unit, as opposed to monad, in other words, one person at a time, in the context of social relationships, which is extraordinarily complicated and really technically challenging, so challenging that very few groups in the entire world have ever tried to take this work on. And yet we believe that social behavior is the absolute foundation of everything that we do in psychiatry. There's very little that we do that that just involves one person in isolation. Problems of living are problems living with other people. And she, in her work, has applied these questions not only to these kinds of limited dyadic equations, but thinks in grand scale about conflict and broader issues as well that you'll hear about. So I think today we're in for a really special treat, and we're thrilled, Joy, to have you come and deliver Grand Rounds today.
Good morning. Thank you, John, for that really lovely introduction. And thank all of you for coming to the January 11 Grand Rounds in Psychiatry. It's certainly my privilege to be here to deliver news of the progress that we've made in this grand enterprise. I want to start with the really big picture here, which is that we are in our laboratory now trying to understand the fundamental mechanisms and principles that guide individuals in interactive behaviors. We're at the very beginning, and so this is a very humble report, but nonetheless, we're getting our money's worth in thrills and chills, as you can imagine. So I want to start with what is absolutely self-evident, and that is we are a profoundly social species. And this is true from the very beginning of our lives, throughout all of our developmental stages, and at the end of our entire lifespan. And yet, as John pointed out, and yet we know very little about these interactive behaviors that guide our interpersonal interactions. The fact that socialness is highly conserved across the animal kingdom also highlights the fact that this is an extraordinarily important question. So today, in presenting our progress to you and our approaches to this question, I'm going to start with a very brief summary of what we really do know about this question as a way of setting a foundation for us. And then, as John promised, and you might imagine, I'm going to tell you about the technical details of functional near-infrared spectroscopy that actually enables us to take measurements of two individuals interacting simultaneously. And then we'll get to the heart of it, but I'm going to introduce two hypotheses, the interactive brain hypothesis and the dynamic neural coupling hypothesis. And I'm going to test each of those hypotheses with respect to two very simple interactive behaviors, face-to-face and talking and listening. And we'll look at both of those paradigms in the context of these two hypotheses. And finally, I will lay the groundwork for what I believe as an emerging theoretical framework. And then hopefully I'll be able to talk about what I think are some exciting applications for this field. So, okay, let's see. Here we go. Starting at the very beginning of what we know, a brief summary of current social neuroscience. Again, this is absolutely self-evident. We're all very aware of this. Faces are important. Now, this has been known for a long time. We go back about 3 decades with Nancy Kanwisher's very seminal studies on faces in the MRI. And in her studies, she presented faces and objects and noted that the activity, the magnitude of the bold signal during a face was greater than the magnitude of the bold signal during viewing an object when one was looking at the inferior temporal gyrus, which is really actually the fusiform gyrus, now called the fusiform face area. By the way, I'm going to go through just a few foundational pieces of information to start this, and I'm sure that almost all of you in this audience are familiar with this. So this will be just kind of a quick review for everybody. It's sort of fun to go back and sort of look at our history in neuroscience a little bit. This is 3 decades old, but a very, very seminal, very important finding. Similarly, about 3 decades ago, around the same time, it was discovered that faces are extraordinarily important as represented by EEG signals. And this was discovered, well, what the finding is that there's an N170 signal that is in response to faces, whereas objects is a later signal with less magnitude. This was studied by Allison Pugh, Spencer, and McCarthy here at Yale. This lovely illustration here was done by Swetha Dravida, one of the MD PhD students in my lab. More recently, the fine-grained coding mechanisms for faces have been worked out in just brilliant detail, I think, by a group at MIT, Chang and So. And what they have done is have, they're doing single unit recordings from macaques looking at parameterized faces, which means that we take details of faces like, say, size of the mouth, distance between the eyes, shape of the brow, and so on, about 50 parameters, recording from the inferior temporal gyrus, and looking at the relationship of spike firing with the magnitude of the parameter related to the faces. And the finding is an extraordinary finely grained responsiveness to an arborization of neurons that are dynamically organized to measure faceness. And the notion is that this system is extraordinarily flexible because there are many neurons responding to many parameters of faces. And this allows a processing system that is extraordinarily rapid and accurate. And that's what we need, is a system like this to explain how faces are so important to us in social interactions. And so this is a very important seminal finding for us as we develop our models for the kind of work that we're doing. For the last 10 years, people have been aware of the fact that there's a sweet spot here in the right temporal parietal junction. This is a summary of about 4,000 studies that was published not too long ago by Carter and Hoytel, showing that all studies that represented things, socialness, particularly theory of mind, showed activation in this area. And this is an area where processing streams from attention, memory, and social factors seem to come together. So the right temporal parietal gyrus is sort of a region of interest that we want to keep our eyes on because of this prior work. And finally, neuroimaging studies provide ample evidence of the canonical systems associated with language, and yet we know very little about the language system in real-life interpersonal communication settings, but we know quite a bit about the canonical areas when people are testing language mechanisms from inside the scanner. And so if you ask somebody in the scanner to name a series of pictures and describe them, it's a very standard task, picture naming and description. What we see is a canonical set of language areas associated with Wernicke's area, mostly associated with representation, and production, Broca's area, and so on. But we know very little about how these regions actually work when we're in an active dynamic mode. So to start our trek on answering these questions, we start with the two guiding hypotheses that I alluded to before. The first, the interactive brain hypothesis, and this comes to us, believe it or not, from philosophers. De Paulo and de Jager, 2012, published this. And the nice thing about being a philosopher is you don't have to have any data. And so, these guys, they get this bright idea and they go out and they publish this thing and everybody loves it, but there's not a single data point associated with it. It's really cool, really cool. Anyway, the notion is that there are that two dynamically interacting brains engage dedicated neural systems that are distinct from single non-interactive brains performing the same task. And what that means is that if you're interacting with another individual, your brain is different than if you're doing the same task alone. Nice idea. Secondly, and this idea comes to us from Yuri Hassan and his group at Princeton, and there is some data associated with this. He puts people in the scanner on multiple times and correlates their signals at different time frames. But his idea is a seminal idea as well, I think. Then that is, we call it the dynamic neural coupling hypothesis. And the idea is that neural mechanisms that underlie social interactions are dynamically coupled. And so we can identify these systems that are engaged by their coupling with each other across the dyad. Okay, so for the next part of this talk, I want to talk to you a little bit about how in the world we do this. Because as John mentioned in his introduction, this is not common practice in neuroscience. And so here we have a setup, sort of illustrating kind of how it works. And you notice that the people are wearing these funny caps. And this funny cap has optodes. They're associated with near-infrared spectroscopy. Now, I just want to point out that, follow up what John said, that before we started doing this, people have done near-infrared spectroscopy. In fact, just one second, I think, I'll come back to that slide in a minute. But this is a historical sort of picture. Again, near-infrared spectroscopy was discovered about 1990, and you can see a steady increase in publications up to the present time. So this is not a new technology. Most of the work with functional near-infrared spectroscopy has been done with infants and people who are studying developmental issues with people that could not go in the scanner, particularly, as I say, particularly infants. Now, if we look down here, the same search engine, and we ask it to add the word hyperscanning or looking at two individuals at a time, you see a very modest number of papers. So if any of you out there have never heard of this before, don't feel too bad, because there isn't very much. In fact, this little incline here, we have a lot to do with that. Let me just go back to the slide where I was. Okay, here we go. So this gives you an idea. This is kind of a new setup. And to our knowledge, this is the largest near-infrared spectroscopy system in the world, and essentially what we have are two essentially full head caps for two individuals. These are fiber optics. Notice the fiber optics leading to a central computer here. And I'll talk about the details of this in just a moment, but I also want to point out that there are a lot of other things going on here, and these other things are very important as well, because now that we are free of the high magnetic field, it's so wonderful, we're free of the high magnetic field, we can do a lot of things that we couldn't do when we had to put people in the bore. And so we have here eye tracking. We've got Kinect cameras that are looking at facial expressions and categorizing facial expressions. We've got, I think you can see an amplifier here. We've got EEG, we're collecting simultaneously. A little analog response dial here for subjects to give us ratings of what they're experiencing, and so on, other physiological measures as well. Okay, so So let me tell you how it works. Basically, we have two types of optodes on the head, emitters and detectors. And these emitters are emitting 3 wavelengths of light. 780 nanometers and 830 are the most important. Note the 780 is designed around the absorption spectra of deoxyhemoglobin, and the 830 similar designed around the absorption spectra of the oxyhemoglobin signal, as you all know, proxies of neural activity. So these wavelengths are emitted, they are diffused in the neural tissue, and they are detected as they exit the surface of the brain. So what we're looking at are oxygen changes associated with task-based activity in the brain. These absorption measures then are converted to concentration, and these concentration measures become our dependent variable. Channels are identified as the space between the emitter and the detector. And I'm sure that from this little diagram here, you can begin to see some of the, let's go back here, some of the limitations of the system, which I will talk about in more detail in just a minute. But our penetration is about 3 centimeters, and it's on the surface of the brain. So there are a lot of things that we would like to look at, like that stuff down under, down there in the hippocampus and the emotional areas of the brain. We're not going to see with this technique. What we see is what's on the surface of the brain, but with our techniques, we can get the whole brain. Our surface resolution is also about 3 centimeters. Primary limitation. However, one of the advantages here is that we're acquiring images every 27 milliseconds. 27 milliseconds. In a scanner, we acquire images about every 1.5 seconds if we're doing it right. So the order of magnitude difference in the temporal resolution with which we acquire these signals, but not spatial resolution. So let me give you an example of what a signal actually looks like. And for those of you who know me in my laboratory, you know that I really like raw data. I like data that hasn't seen the light of day of a computer, that we see it just as it comes off the machine. As it's collected. So here we have a participant, and this arrow is indicating that I'm going to look at the signal from 1 optode. This is 1 optode in 1 trial. And one of the nice things about near-infrared spectroscopy is that we collect 2 signals, the oxyhemoglobin and the deoxy, and I'll show you both. The task is going to be finger-thumb tapping, and we're going to be doing it in 15-second intervals. So we tap for 15 seconds, rest for 15 seconds, tap and rest, the standard alternating rest and tap that you would see in the standard fMRI scanner. And so here we see the signal. This is the oxyhemoglobin signal, a well-behaved, bold signal. It takes about 8 seconds to reach its max. It hangs around while you're doing the task, and then when you rest, it falls down. It takes it about 15 seconds to fall down to where it's at its baseline again, and so on and so on and so on. A well-behaved, bold signal. Now, the deoxyhemoglobin signal is acquired at the same time. We tend to use the deoxy signal rather than the oxy in our studies because it is the deoxy signal that, of course, is most closely related to the fMRI signal, and it is the one that is least confounded by the systemic effects such as blood flow that's not related to neural activity. Okay, raw signal, and that's how it works. It works as it should for a well-behaved hemodynamic signal. There are a few other details, a couple of which I've already mentioned here, spatial and temporal resolution, and that we're sort of unique in the field by the extended coverage of both heads. This is an example here of a full head coverage where we have both fNIRS and with eye tracking, with the EEG, the facial classification camera here, the analog responses and respiration monitors and so on. And you can see we need these other measurements because you'll see as we get into the complexities of trying to understand what's going on behaviorally between two individuals so that we can relate that to the neural systems, we need all of these measures. So this is just an illustration of the optodes in green, and I've put in, we have 32 EEG channels that are interleaved for many of our studies in this layout. Okay, so next part of the talk. So what do we do with all this technology, and how do we make it tell us something about two brains in interaction? So let's start with this interactive brain hypothesis. And basically, as I said before, what it means is that you've got two brains doing a task alone. They're going to be doing it differently than if we've got two brains doing it together. And so let's see if we can lay any numbers, any objective quantification to this idea to see if it makes any sense. Isn't that a wonderful picture? I love it. It's you don't need to say anything. Eye contact matters, doesn't it? You could write a book about that. Anyway, so we take what I think is one of the most fundamental behaviors of interpersonal interaction, eye-to-eye contact, and we can adapt it to the interactive brain hypothesis and say that we think that eye-to-eye contact is going to engage the brain differently than if one is looking at, say, in this case, a video of a face or a picture of a face. And so basically our paradigm is we have people looking at each other face to face, and then mutually looking at either a dynamic video face, which is this is the data that I'm going to show you, because the dynamic video face is is probably the better control because of the movement of the face than the picture face. But either of these controls can be used. And so our basic paradigm is this, that we, activity, 15 seconds, rest, 15 seconds, activity, 15 seconds, you get it. But in the activity period, people look at each other for 3 seconds and then they look away. They divert their gaze 10 degrees. The reason for that is that you can't just stare at somebody for 15 seconds. You look for 3 seconds, you look away, you look for 3 seconds, you look away. And we treat this whole activity block as a whole block, but it's just done in mini epochs, just because that's the way the system is. So the first findings that I want to show you are these, and it's pretty cool. This is the group data from, we have 30 people here, group data of the looking face-to-face, we call it interactive gaze, versus looking at the gaze of the moving face video. And you see here we get lovely high-level visual activity as one would expect, but look at what we have here. We've got that plus angular gyrus and fusiform gyrus, loud and clear. And so our findings basically are consistent with the interactive brain hypothesis. And there's a suggestion here that there's sort of back of the brain. Remember I showed you the right TPJ? This is the right temporal parietal junction. It's a little bit excited by the face-to-face as opposed to the dynamic video. So nice finding. Now, we test this hypothesis with another paradigm, and that's with talking and listening, because it's another one of our really fundamental behaviors for interacting with each other. And the way we do this is that we have people talking and listening without interaction. Now, all of you who are parents who know perfectly well what it's like, you can talk and nobody listens, right? You know what it's like. You talk and nobody listens. Or you talk, people listen, but they don't respond. You might as well be talking to yourself. Okay, so it's something like that. But we've developed the picture naming and description task, and I will say a little bit more about that in just a minute with this paradigm. So here in this paradigm, the timelines are going down. We have two people, and they're taking turns. They're, again, 15-second epochs of time. So he talks, she listens, she talks, he listens, and we go down for the 3-minute run. So we do this talking and listening in two types, one without interaction. And so the picture naming and description task goes something like this. He gets his picture, it's a clock. He talks about it for 15 seconds. She listens. She gets her picture, say it's a camera. These are just random pictures. She talks about it for 15 seconds. He listens, doesn't respond, and so on. But now, we can do this same task in a different way. We call it the dialogue. And in this task, we start the same way. But instead of talking about the camera at first, she responds to his comments about the clock. So in each interaction, there is a response from the previous person's narrative. So it's interactive. It's minimally interactive, but it's interactive. So, okay, the reason that we chose this task is because we really do know the answer. And the reason we know the answer is because this object naming and description task has been used for so many years as a mapping technique for neurosurgical planning. So that for someone who is planning to go to surgery, we can map their Broca's area and Wernicke's area just doing the object naming and description task in the MRI. And of course, we expect these canonical areas. And so being kind of a conservative, cautious scientist, what we do is before I start analyzing the data, comparing dialogue to monologue, I want to be sure that we've got a task that really works. And so what I did was I combined all of the data from both of these conditions, and I asked, do I see these canonical language areas again? And so the answer is, yes, we do. We look at the talking greater than listening comparison in red, and the listening greater than talking comparison in blue. And we recapitulate the articulatory, the productive systems, and the reproductive system. And I do it in two ways. We can analyze the individual channels, or we can do it what we call voxel-wise. And this is what most people do. We pretend that we're doing MRI. And so what we do is that we pretend that we have pixelated the entire brain with voxels about the size of a grain of rice, like what we do with MRI. And then we analyze the data that way, and you can see if we superimpose them, we recapitulate what we know is true. So now, with this, we can feel pretty confident in our analysis as we compare the monologue talking and listening with the dialogue talking and listening. So we start with two brains and our two conditions, and I'll show you the data. The first set of data I'm going to show you is just the talking data. Monologue, dialogue. Doesn't look like we support the interactive brain hypothesis here. So much for the philosophers. Wrong. It doesn't. Your productive system just doesn't care whether anybody's listening to you or not. It just does what it does, okay? But this other receptive system cares a lot. You can see that in the monologue mode, a superior temporal gyrus, just exactly what you expect, but the region that is active during the interactive mode is not only increased in magnitude of activity, but also the constellation of areas now includes not only the superior temporal gyrus, the middle temporal gyrus, the angular gyrus, and the supramarginal gyrus. So we support the interactive brain hypothesis with the Wernicke's area. And I want to point out that Wernicke's area is the left side homologue to the right temporal parietal junction. It's the same deal, it's just on the other side of the brain. So again, I like to think of these things as back of the brain functions. And so with both of these paradigms, the talking and listening and the face-to-face, we have some evidence that interaction matters to the brain. So how does it work? What is going on? And so for that, to get a little bit closer to the answer to that question, we turn to the dynamic neural coupling hypothesis. And the idea is that in two brains in a dyadic situation, when individuals are actually engaged in interaction, that the neural systems are actually coupled in some way. So let's look at how we test that. The correlation between activity in two brains is really just the extent to which the signals are correlated. And we use wavelet analysis for that. So the situation is something like this. You get a signal from some place in one brain, complex signal, and you get a signal from some place in another brain, and you ask, are they correlated? Here we've sort of drawn it as they are. They can also be completely uncorrelated. And the synchronous or not synchronous relationship is often measured by wavelet analysis, and as do we. So let me show you how this works and why. So the classical method for measuring cross-brain coherence, something like this. You take the channel constellations of the brain. I sort of divide them up into areas that are anatomically homologous. Wouldn't have to do that, but by doing this, we minimize the multiple comparisons problem a bit. And for those of you who do fMRI, know that that's a pretty serious problem. And so this is one of the ways that we can deal with that to some extent. We take a signal from a given area on both brains, and this signal is decomposed. And instead of decomposing it like one would do if you were doing a Fourier analysis where your decomposition is continuous, we decompose it into its wavelet components. And so the wavelet components are, of a complex signal are illustrated here. These are the high frequency components and the low frequency components. And by the way, this is a technique like psychophysical interaction or PPI interaction, where we do functional connectivity and functional MRI, we actually take out the main effect. Well, this is exactly the same thing. We take out the main effect by doing this decomposition with wavelets. And so what this means is we've got high frequency stuff here, low frequency stuff here, and in this area here, we've got a sweet spot of frequencies based on what we know of the well-behaved bold signal, where we expect the correlations to be. We expect them to be between, like, say, 6 seconds and maybe 15 seconds, something like that, sort of at the tail end of our hemodynamic cycle. So we expect to see correlations in this area, on this time frame of the wavelet decomposition. So this is what we do. Let's just take an example of the data from the face processing that I showed you earlier. And we take the two dyads as a group. Remember, we have 30 people here. We plot on this axis the correlation between two brains, and we plot the wavelet decompositions here. Notice this is the wavelet region of interest, where we know where our bold signal is the most significant for us. And what we see for two brain areas, angular gyrus and fusiform gyrus, a very significant increase in correlation between the two brains in this region. So what this says is that this cross-brain coherence exists in areas of interest that we saw with the contrast that I showed you earlier. And it's highly suggestive that we've got some support for the dynamic neural coupling hypothesis. And one looked at this, if you wanted to see a diagram of this, we've got this fusiform guy and the angular gyrus guy, and these two guys are going back and forth during face-to-face interaction that is not happening when the two people are watching the video. So we can interpret this as being consistent with the dynamic neural coupling hypothesis. Now, we test this in another way, and this was done by another student in my lab, Matthew Piva, and it turns out that we did this test before we actually did the face-to-face experiment, or I guess they were kind of going on simultaneously, but I didn't think of one as being a test of the other, and yet in retrospect it is. So consider this a test of the angular gyrus to fusiform gyrus neural coupling model hypothesis. And so when we were doing this experiment, we didn't have the presence of mind to take a picture of two people sitting in front of each other playing poker with optos on their heads. But you guys can get it, right? So we borrowed this from, I hope it's legal, from somewhere on the internet to illustrate that people playing poker. And I want you to note here that these guys are well aware of the fact that faces really matter, and they're trying really hard to keep people from seeing their faces. So in this experiment, the students were playing poker. It was a simple single trial poker where you folded or bet on each trial. And you were playing, and sometimes with a human as your component, that is the person sitting across from you, or both of you, although sitting across from each other, were playing against a computer. So the data are very interesting, that in both cases you have faces in front of you. But in one case, the face gives you information about what you think your opponent is going to do, although they're trying not to share that with you. But nonetheless, there's information there. And in the other case, you're playing against a computer, there's no information. So again, what we see is a very strong correlation across the angular gyrus and the fusiform gyrus of the two brains playing poker during the human-to-human condition as compared to the human-to-computer condition. Always, I just want to point out here that in all these cases, we do the scrambled partners control, and this convinces us that what we're seeing here is due to the individual dyads and not the group process. So again, support for this angular gyrus to fusiform network, if you will, have associated with interpersonal interaction that has to do with people looking at each other. So basically, this is what we think happens, something like this. He looks at her. His brain registers her face. She looks at him. His brain registers her face. Her face registers his brain. Yeah, right. Okay. And so on and so on. So this goes back and forth and back and forth and back and forth. And so the angular gyrus, the fusiform gyrus are playing a part in this. So what's going on? And so what we think is happening, of course, there's all these things going on: facial features, facial expressions, gestures, rapid eye movements, stuff going on under the hood that's being processed by these guys. It's going very rapidly. And so this is where our current work is focused. I want to go on with some other points, but I want you to know that we're into figuring out what these guys are doing in this active interpersonal interaction, and we think it has something to do with this, and that's why in our experimental paradigms and in the situation why we have all these face cameras, the timing stuff going on, the EEG, all this other stuff going on, because this is what we're trying to figure out, because we're working in this context that we've set up. Now, let me show you that again similarly for the talking and listening, where you have a what I think of as now a linked language network that involves the superior temporal gyrus and the subcentral area. These two areas that are correlated between the two brains. One would expect, of course, the superior temporal gyrus. That's Wernicke's area. We saw that in the contrast. Does anybody in this audience know anything about the subcentral area? It happens to be Brodmann's area 43, and it's hardly been mentioned in the literature since Brodmann in 1909 wrote about it in his description of the cytoarchitecture of the area. And actually, subcentral area sits right here, sort of at the foot of where the right across the central sulcus of the posterior aspect of the sensory system and the motor system, although Brodmann says that it really is part of the posterior of the brain. It's behind the central sulcus. But it seems to be involved, we see it quite a bit, it seems to be involved in the interaction having to do with language. And I have a feeling it has something to do with stopping and starting these interactive processes, but I'm not sure. But we're pretty sure what we know what the superior temporal gyrus does. But nonetheless, in this language back and forth, when there's dialogue, there is more coherence between the brains than when there is the monologue condition. And that's really pretty cool because the two brains are doing different things. Somebody's talking and somebody's listening. It's not like you're just doing eye-to-eye contact where everybody's doing the same thing. These are two different things, and yet the brains are quite correlated when there's an active interaction going on. So basically the system is this. He talks to her, and it registers in her brain. She talks to him, it registers in his brain, and we think it's these two areas that are very involved. Of course, there's probably other areas, but right now we're pinning it down to what we actually see. And the language system is doing a whole lot of things other than just understanding the meaning of language. The word features, acoustic expression, intonations, and nonverbal utterances and so on, which is why we're actually engaged in looking at these particular variables in the context of interaction. So we've got an emerging theoretical framework. Again, I want to point out that this is a humble beginning. This is really the beginning of what we think is a theoretical framework. We think we have evidence to support that live social interactions recruit specialized neural operations. And I'm not sure what I mean exactly by specialized neural operations. It could be that we mean that the neural operations are distinct, different. They're not involved at all in the task in solo mode. I don't think that's the case, but we could mean that. We certainly mean that the specialized operations have a greater magnitude than other operations, than solo operations. So we'll do a little bit of fine-tuning and shaping of this idea, but this is the beginning of the idea, that neural mechanisms of interaction are dynamically coupled. And we're really interested in this hypothesis because we'd like to know what's going on in the coupling. And one of the main hypotheses that we're working on now is that we think when neural systems are coupled, they're linked together, that they're actually sharing information. And the sharing of information is something that we can test. And so we've got some tests going on asking about if giving people information via the coupling, do we see the expected modulation in the coherence? And so these are pretty active areas of research. So now, at the end, why in the world would perhaps people in psychiatry be interested in this type of work? Well, maybe you're not, but you could be. And a reason why I think you might be, or a reason why I'm interested in this, is that just as John said in his introduction, that psychiatric conditions may be modeled as disorders of social interaction, and that understanding these models better might lead to a new focus for diagnosis and intervention. And I want to take this idea just a little bit closer to home because I have kind of a neat surprise on this. One of my medical students in the lab, Rahil Rogiani, sort of took this work to heart and decided that he wanted to study. He actually came to us wanting to study music in the brain, but we didn't quite do that, but we almost did it. So he borrowed our talking and listening paradigm and had people drumming. See the drums here? Two people singing. We had the presence of mind to take pictures of this. I don't know why we didn't with the playing poker. But anyway, so everybody that participated in this study got little lessons in drumming. And what we asked people to do, this sounds a little bit crazy, but it was so neat. We asked people to, we gave them pictures, but these pictures were calibrated on the IAPS library. The IAPS is the International Affective Picture System, and these pictures are calibrated with respect to valence and arousal. And so we gave people these calibrated pictures, and we asked them to drum, just to use their drums to tell each other what they felt from those pictures. And they got really good at it. It was really, really cool. So just drumming back and forth and back and forth. Rahil published this recently, actually. It was kind of neat. Now, there are many findings in this study, but I just one of them that's relevant to my talk today is that when we look at the equivalent, remember the talking greater than listening in the previous paradigm. This is drumming greater than listening, and you can see exactly what you expect. This is sensory system. They're drumming with both hands, bilateral sensory. So makes sense, all right? Good. I love it when data makes sense. And of course, listening and drumming. Now, I thought that we might be activating Wernicke's area, because it's kind of a language, right? But no, the right temporal junction wins. And what this means is that as people were drumming, they're not using words, but they're communicating with each other something that has to do with emotion. And so we thought this was a very important finding. But my student, Rahil, doesn't stop at that. And he says, all right, this might have something to do with something related to, that could be related to clinical service. And so he goes to a halfway house here in New Haven, and he sets up a drumming group and has a control. There are people that are treated as they normally treat in this halfway house, normally treated, in this halfway house procedure, and those that are not. And in this halfway house is a place where men come as they are in this period of reintegration back into the community following incarceration as adult men. And so he has this drumming group, and he has a drummer come in, teach people how to do it, and they begin periodically, I think it's weekly, talking to each other with drums. All of those people stayed in the halfway house program until the end of their series. Those that were treated normally didn't complete the process. So this is descriptive, but I hope it encourages you to think a little bit out of the box. That sometimes there might be something here that would be really that could be integrated at some point. But nonetheless, for sure, we would all agree that understanding the principles of interactive behaviors could be important in treating them. Secondly, and this is a little obvious, that social defects are the hallmark of autism spectrum disorder. And we're engaged now in a very active and large study in collaboration with Jamie McPartland in the Child Study Center to look at the neurocircuitry that is engaged in autistic individuals during eye contact in live interactive conditions. And so we have a setup very much like what you saw, but we use a confederate as the partner. And these, so far, the preliminary data from this study are extraordinarily interesting. You have to invite me back another time to tell you what it really is, but this is a very successful project. Finally, and this again, I'm going to end with the self-evident that social interaction is a significant foundation for well-being and longevity, and benefits for understanding these neuromechanisms go a long, long ways. And here I've just mentioned a few, but prevention of depression, loneliness, and perhaps even maintenance of good health practices. And we have some ideas. Now, this is something that we haven't done, but I could hardly wait to do it. We actually want to start thinking about ways to design companion robots, knowing a little bit more about the human responsiveness to interaction behavior. If we could help design the behaviors of robots that could enhance the experience of individuals interrelating with companion robots. And finally, and this is so much fun, that we've actually made some progress on this one, use of therapy dogs. Wouldn't it be cool to understand how we communicate with across the species? For example, your dog or your cat. You know perfectly well you have a relationship with them, right? And wouldn't it be wonderful to understand a little bit more about those bonds and how we might actually use them in conditions of enhancing relationships with both the dog and the master. Okay, so those are my ideas. The people that do all the work, and here they are. First of all, I want to point out the mural here. By the way, I know we have a committee somewhere for art in the medical school. Well, I have an idea. Why don't you just employ students to go out and paint some really cool pictures about what they really do and put those up on the wall. This is a mural in my lab, and this is student Jenny Park, who was both a neuroscience major and an art major. And her mandate at the end of her senior year was to paint a mural of what Yale had meant to her. And so here it is. And I'll introduce these people in this mural with the people. But people that really do the work are Ray Capiello, our manager, lab manager, Swetha Davida, MD, PhD student, Chen Zhang, computational neuroscience. Jenny has graduated, the author of this wonderful mural. Oh yeah, I'm there. Adam Noah, a research scientist, a computational engineer. Yumie Ono, sabbatical visitor. Jen Kuzukreo, research assistant. And not shown here are Courtney DeCoco. Courtney is a post-bac assistant. And Megan Kelly, graduate student. And Rahel Rogiani, a medical student. This mural is in our lab. And so, by the way, we always need participants. And so if you come and visit me in the lab, we might actually ask you to put on the optodes. So be warned. But other than that, you're totally welcome. Thank you.